Okay. Once again, we're here to do the review for the questions of Form 6, Paper 1. And today we are continuing with question number 7 and this review of the paper 2019. And the question number 7 says this. Okay, the question says, why does transpiration mainly occur through leaves and not so much through the cuticle or lenticel? But we summarize the properties of xylem, which makes it suitable for the long distance transport of water. Okay, let's start with 7a. Okay, which says why transpiration in leaves? Why transpiration mainly in leaves, okay, and not in the cuticle or lenticel. The reason is that there are two reasons. The first one is that the leaves contain very large stomata number. It contains leaves large number of stomata. This one allows the movement of air vapor outside of the leaves compared to the cuticle or the lenticel. The second is leaves has large surface area. Okay, so larger surface area, the higher rate of transpiration. So the larger surface area, these 2 points, the larger surface area, the larger the rate of transpiration. So each of these 2 marks, total 4 marks. Part B, the properties of xylem. The property of xylem for its function. Okay, this is like adaptation of xylem to the function it performs. We know that the xylem transport water and mineral salt from the roots to the different part of the plant. Okay, the property or the adaptation, it has a long tube. Okay, it has first one long tubes. Okay, these tubes whereby the fusion of the neighboring cells occur with the breaking down of the cross wall between them. Okay, long tube. Okay, with breakdown. Okay, breakdown of cross wall. It means it does not have the cross wall, so it allows the water to move freely. This is one. The second one, adaptation of the xylem, is that no living content. Okay, no living content. Okay, this one consists of the dead cells. Okay, so because of no living content, so there is less resistance to flow of materials. Okay, if there is a living content, it will prevent movement because it is selective permeable. Since this one does not have the leaving, it is free to move. Another is that the tube are rigid and do not collapse. Okay. Number 3, tubes are rigid. Okay, it is lignified. Okay, it is lignified tubes. Okay, which contain the lignin material, which prevent from collapsing. Okay, so it will transport material without collapsing. Okay, another adaptation of the xylem Okay, adaptation number 4 of xylem, okay, is that the xylems has fine tube necessary to prevent a water column from collapsing. Has fine tube, okay. This one tubes preventing water column, okay, water column from collapsing. This is capillarity. So it allows, due to the small diameter vessel, it allows the water column to continue up to higher parts or different parts of the plant. So that is adaptation of xylem. So it has 4 marks. Each of this one, student, you are supposed to answer only 3, and each one contain 2 marks. So total of 6 marks from there. Okay, question number 8. Question number 8. Okay, the question says, state from the it says, state the significance of double fertilization in flowering plant. The second, part B, it says that describe the process of implantation and formation of the placenta. Okay, the first question, question number 8A, state the significance of double fertilization. Part B, it says describe the process of implantation and formation of placenta. Okay, 8A, okay, which says significance of double fertilization. Okay, the first significant of double, first of all, double fertilization, this is fertilization whereby the generative nuclei of pollen grain divide mitotically to produce 2 male gametes. Gametes on which one fuse with the female gamete or the ovum and forming a diploid zygote, and other fuse with diploid nucleus to form a triploid nucleus known as primary endosperm nucleus, to form a primary endosperm nucleus. That is, it continued to form endosperm. Okay, the significance of this one. Okay, significance. The first one is it brings recombination of character, okay, resulting to a variation of offspring. Okay, it bring, this one bring, okay, recombination, okay, recombination of character, of character. This bring, okay, bring the combination of character resulting to variation in offspring. So offspring. So we know that it is normal fertilization between. Fertilization from different organisms, which is sexual reproduction, which bring variation. Another is that the second is it gives rise to new plant. Okay, another significance, it gives rise to new plant. Okay, it gives rise to new plant. How? This is because formation of diploid zygote, which develop to embryo, then the plant. Okay, whereby it starts from zygote. After fertilization, zygote is formed, then embryo and a new plant. Okay. Number 3, dear viewers, okay, is the significance of double fertilization. It restores the diploid condition, restore the diploid condition of the plants, okay, by fusion of haploid male. This one is by fusion of haploid male and female gametes. Okay, so it helps to maintain the diploid condition, whereby the haploid male and female gametes unite to form a zygote, which is diploid. Okay. The fourth one, the significance is that, okay, this one, it results to the formation of triploid primary endosperm. Formation of triploid. Okay, triploid. Formation of triploid primary endosperm. Okay, endosperm nuclear. This nuclear develop to endosperm for food storage. Okay, in the seed. Okay, food storage in the seed. So that is number 4. Okay, so it forms a triploid primary endosperm nuclear, which develop to endosperm. Okay, then lastly, number 5. Okay, this one, the significance of double fertilization is that it gives stimulus to plant, due to which ovary develop to fruits and the ovule develop to seed. Okay, it gives stimulus. Okay, a stimulus to the plant, whereby ovary develop to fruit and ovule, okay, this one develop to seed. Okay, so without this fertilization, that stimulus will not be there, whereby the ovary will remain as ovary and the ovule as ovule. Because of this fertilization, so it makes the ovary to develop to fruit and ovule to develop to seed. So that is question number 8A. So if you answer all these, you're supposed only to answer only three. Each one, only three among this one. Each one has 1 mark, so total of 3 marks. Okay, from there. 8B. Okay, 8B, it says, describe the process of implantation and formation of placenta. Okay. Implantation and formation of placenta. Okay, so this implantation occurred and formation of placenta, it starts by, after fertilization, the zygote moves through oviduct and while it undergoes cleavage to give rise to blastocysts. Okay, so zygote, this one moves through oviduct, and at the same time, while it's moving there, the cleavage occurs, okay, to give rise to this one to structure which is known as a blastocyst. Okay, a blastocyst. This blastocyst has 2 layers. Okay, so blastocyst has two blastocyst has 2 layer, which is outer layer of cell, which is known as a trophoblast. And the inner part, which is known as a cell mass. So that is how the zygote developed to blastocyst, by cleavage while it passes through oviduct. And this blastocyst has 2 layers. That is outer layer, which is trophoblast, and inner part, which is called cell mass. The second, under this one, as you explained, so you have to write that way. Okay, after 6 to 10 days, 6 to 9 days after fertilization, the trophoblast make contact with endometrium of uterus and embedded through it. Okay, so 6 to 9 days, okay, after fertilization, okay, trophoblast, or the outer layer of cell, okay, come into contact with endometrium, okay, endometrium, okay, of the uterus of mother's uterus, okay, and embedded, and this one, and embedded into it. So this entering into endometrium of the mother's uterus is called implantation. Okay, this is implantation. So the third process is that after implantation, the trophoblast differentiate into two layers. Okay, after implantation, trophoblast, okay, differentiate, okay, into two layers. Okay, two layers. Okay, this differentiate. The outer layer forms a trophoblast villi. Okay, the outer layer forming trophoblastic villi. Okay, trophoblastic villi, and later this trophoblastic villi, okay, grow into endometrium. Okay, which grows into endometrium, or the uterus, and later will form inner layer called corium. Okay, this will not be formed now, it will form later. So there are two layers. The first one is this trophoblastic villi, which grows into endometrium, and later that inner layer, it will form the corium. That is later. Number 4 process as it continues is that, okay, the space between trophoblastic villi and endometrium, it forms a cavity. Okay, space between endometrium and trophoblast villi forms a cavity known as a cavity known as lacuna. If they are plural lacunae, so it forms a lacuna, a cavity it forms a cavities because there are many cavities known as a lacunae. This which makes the endometrium spongy, which make endometrium spongy. It makes it soften it and make it spongy. Okay, so okay, so number 5, Okay. Number 5 is that the hydrolytic enzymes released from the villi, trophoblastic villi, causes the blood vessel in the endometrium to break down and fill the lacunae. So, number 5 is that enzymes released by trophoblastic villi. Okay, this villi, it releases a certain types of hormone which causes the blood vessel to break down. Okay, causes blood vessel. Okay, blood vessels to break. Okay, the blood vessel to break, and blood fills the lacunae. Break and blood fills the lacuna. Okay, from there, now the exchange between this allows the exchange of material. Okay, at the same time here, meanwhile, the trophoblast continue to grow to form inner layer, which is chorion. Okay, here, trophoblast continue to grow to form chorion. Okay, this is the place where I say that later the inner layer of the outer cell that is trophoblast is chorion. This is the time where it is developed to inner layer. Okay, and number 6 is that the outgrowth from the embryonic hindgut come into contact with chorion, which is highly vascularized to the formation of large chorionic villi to form a placenta. So there is an outgrowth of the outgrowth from embryonic hindgut, which come into contact with chorion. The chorion, which is the inner layer of the outer layer of the outer layer of trophoblast. So this is inner layer of the trophoblast. So there is that development of embryonic hindgut, which outgrow and come into contact with chorion, which is highly vascularized, and which is highly vascularized, or it contains a blood vessel, okay, for transporting of material to the formation, vascularized to the formation of large chorionic villi to form a placenta. Vascularized, okay, to the formation of chorionic villi. Chorionic villi, the chorionic villi which develop to placenta. This is how the placenta is developed. So this hind gut, there is that outgrowth which come into contact with chorion, which is highly vascularized, to the formation of chorionic villi, which is so long that it forms the placenta. This placenta now take over the role of exchange of material between fetus and mother. This one, placenta, takes over the role of exchange of material between fetus and mother. So the student the candidate you can answer without even putting this number. You can continue. By putting the number is to show you the main point which you're supposed to write, As you answer those 4 points, you're supposed to answer at least 6, especially the first 2, 6. Each one has 2 marks, so the total of 12 marks from there. So that is how the process of implantation and formation of placenta occur. Question number 9. The question says, explain how the eye is able to accommodate near and distant objects. Part A, question number 9A. How the eye is able to accommodate near and distant objects. Part B, in human retina, there are approximately 150 million sensory cells, 7 million cones, and the rest being the rods. There are only 1 million fibers of optic nerve. Roman 1, what implication of those figures will, with the aid of the simple label diagram of human retina, explain the structural basis of the visual acuity? 9A. So to accommodation. Let's start with near object. Near object. In the near object, the ciliary muscle contract. First one is a circular ciliary muscle contract. As this one contracts, the ciliary muscle, this one leads to increase of tension, okay, increase, okay, it increases tension of suspensory ligament. Of suspensory ligament. Okay, and as it increases the radius of curvature of the lens increases. Okay, radius of curvature. Okay, due to the contraction of the ciliary muscle. Okay, to increase refraction of light. Okay, this leads to refraction of light. Okay, refraction of light to the lens. Okay, so it bends more light in such a way the image is focused to the retina. Okay, to the lens. Okay, in such a way, the near object image, okay, is focused to the retina. Okay, that is how. So the ciliary muscle contract, suspensory ligament, also the tension increases. The radius of curvature of the lens also increase. This causes the light to be refracted in the there is greater refraction of light in such a way the image of the near object will be focused in the retina. Part B. Okay, the far object. Okay, the far object, it goes, or the far or distant object, it goes like this. The ciliary muscle contracts. The ciliary muscle, okay, this ciliary muscle here relax. Okay, different from that there. So it increases this one as it is relaxed. The tension of the suspensory ligament, also the tension of suspensory ligament, and here the distance of object is it increases the tension of suspensory ligament. Okay, so and here, okay, so here on top, it, okay, sorry, here on top, here it decreases, okay, tension, okay, of suspensory ligament, and the radius of curvature, that one increase. And here down of the far object, it increases the tension of suspensory ligament, okay? After increasing the tension of the suspensory ligament, what occurs is that the radius of the curvature of the lens increases. So radius of curvature of lens, the radius of the curvature of the lens here, okay, increases. Okay, the radius of the curvature over there, it increases. Okay, it increases, and here, while here, the the radius of the curvature, sorry, here, it decreases. Okay, I'll mix it. Okay, so there on top, the radius of the curvature decreases. Here down, the radius of the curvature of the lens increases. Okay, in the it increases. So when it is increases under this part, refraction is reduced. And the parallel rays of light from the distant object are brought to focus to the retina. So, because now the radius is elongated due to the increase of the curvature, okay? The curvature is like this one. So the radius increases. When the radius is reduced, the lens become like that. So the radius of the curvature here increases. So if the radius is increased, the refraction is reduced here. Here on top, refraction is increased. Okay, this leads to refraction of light to the lens. Okay, lead to this one, lead to increase. Here, increase. Here, the radius of the curvature of the lens increases in such a way, refraction of light here, okay, decreases. Okay, it decreases, and the parallel rays of light from the distant object are brought to the retina. And parallel rays, okay, rays of distant object are placed into retina. Okay, why it is parallel rays is because the distance object, it is very far, so the light will come as parallel. The near object, there is that the bending of the light, okay, toward the person who is looking to it. So the distance object, the light comes like a parallel. So this one, refraction of the light decreases due to the curvature of the lens, due to the curvature of the lens which increases. The lens look like this one. Okay. That this is how the accommodation of near and far object or the distance object it is seen. Part B, 9 part B, it says in human retina there are approximately 150 million sensory cells and 7 million cones, and rest being rods. There are only 1 million fibers of optic nerve. Roman 1, what is the implication of this number? It means that the sensory cell, there are so many. 9 B. 1, this million sensory cells and 7 million cone. 1 million optic fiber nerves. Okay, Roman 1, it says what is implication of this figure? Okay, implication of the figure. Okay, this figure says that, okay, the several sense cells are connected to the single sensory neuron. Okay, several, okay, sensory cells are, okay, this one are connected. Okay, several cells or several sensory cells are connected, okay, to the single neuron, are connected to the single neuron. That is why the neuron are filled, and this one, okay, which increases sensitivity, okay, sensitivity. This increases the sensitivities of the neuron. It increases the sensitivity of neuron. Okay. In the this can be example. This one can be shown in the example of many rods which supply a single optic neuron. This is called convergence. Okay. Example of this is, okay, in rods. Okay. In rods, in the eyes. Rods, okay, in the eye. Okay, in the eye. Rods in the eyes, this is sensory cells. Okay. The rods in the eyes supply a single optic neuron. This is called convergence. Okay, rod in the eyes. So several rods. Okay, the several rods. This one supply a single optic neuron. Supply a single optic neuron. Single optic neuron. Okay, if we have the diagram, you can see it very clearly how this one occurs. And likewise, the combined effect of the simultaneous stimulation of the several cells is cumulative. Okay. Okay. Likewise, continuous. Okay. The combined effect of simultaneous stimulation of sensory cell or sense cell. Okay, that one. Okay, stimulation of that one is of sensory cell is cumulative. Okay, is cumulative and produces, okay, and produce effect to optic neuron is cumulative and produce effect to optic neuron. Okay. In the diagram, you will see that the rods, there are like 3 rods, which their names is connected to the single synapse. Okay, which is connected to the to one bipolar neuron and connected to the optic fiber. So that cumulative, okay, the summation of 3 rods which comes together and pass the information to single synapse. So when you add together, okay, the stimulation, it produces the stimulation which is enough to produce, to increase the sensitivity of, to increase the action potential and the sensitivity for the stimulus to be passed. So that is known as the stimulation which is cumulative, which produces which is produced to the optic neuron, which is then transported to the brain to be interpreted. So that is the implication under here. Okay. Part B. Nine B. Okay. The structural basis for visual acuity. Visual acuity. The visual acuity is a measure of finest detail which unaided eyes can distinguish. So the visual acuity, this is measure of finest detail okay, which unaided unaided eye can distinguish. Okay. The visual acuity in human is due to anatomical arrangement of cones in the retina. Okay. Acuity is due to arrangement of cones, okay, in the retina. This cone in the retina is arranged in such a way that it is highly packed. Okay, it is highly packed in the retina, and not only that, there is absence of convergence. Convergence, that is the cone. Usually, the cones does not have those neuron which occurs together to convert and becomes together in the single synapse. So there is absence of convergence and it is highly packed in the retina. In the area which is called fovea. Okay, so that is why the visual acuity is in that. So nearly 95% of 7 million cones occurs in the fovea. Okay, fovea of the retina. That is why there is that visual acuity. Okay. So with the diagram to illustrate how retina shows that visual acuity. Okay, the diagram of retina. Okay, the retina. the following diagram. Okay, this one comes here. So okay, this one is connected here. And from here, this here, okay, these are the rods. Okay, these are rods in the retina, and this is nucleus. Nucleus, and here nuclear, and from there in the nuclear there is this bipolar neuron. And same this one. Okay, here these are mitochondria. So this part here there is known as the outer segment. Here to this part is known as the inner segment of the retina, and this part here is known as a synaptic region, because there is this synapse here, synaptic region, and this low part here is known as inner surface. So as you can see here, there is this convergence of the neuron which comes to form one, which is connected to one bipolar neuron. And this act of connecting it together, it is known as convergence. While in the cones, there is no that connection of those, the neuron. Only there is the cones is connected to single bipolar neuron, which transmitted the stimuli to the brain or to the optic nerve. So this is how the diagram of retina to show the visual acuity and also to show how it is working.